mutated because it's the conserved region. And these nonsense mutations will cause the truncation of the protein product of a GDAP1 gene from the A here, which is 358 amino acid, to become a 72 amino acids protein, which is totally non-functional at all. Okay, so upon the findings, GDAP1 is mainly expressed in nervous tissue, and it plays a very important role in mitochondrial functions, such as the ones as shown here. And mutation in this gene leads to various forms of Charcot-Marie-Tooth diseases. As shown in this slide, there are two types of Charcot-Marie-Tooth diseases have the mutation and the same gene, which is a GDAP1 gene. Okay. What is Charcot-Marie-Tooth? So Charcot-Marie-Tooth is an inherited peripheral neurological disorder which characterized by distal wasting, weakness, and sensory loss. So what happens is that you progressively lost your sensory you progressively lost your muscle mass and sensory from the extremities towards your torso. So patients carrying this truncation mutation usually will become a wheelchair user before the age of 40. Okay, to verify the diagnosis of the nonsense mutation as shown in the trial exome sequencing and sequencing, they perform electromyography and nerve conduction velocity to confirm the findings. And then these findings has confirmed the test that proband and the younger brother has weak or even no sensory response that can be detected. And from here, based on the clinical features of the patient, with the nerve conduction velocity, electromyogram, and the mutation screening from trial exome sequencing, they finally give a diagnosis for this patient that they have Charcot-Marie-Tooth disease type 4A. Okay, and then this mutation was not previously being reported in any of the studies previously. Okay, so in conclusion, there are more than 90 genes that are known to be associated with Charcot-Marie-Tooth. However, more than 50% of the infected individuals don't know what is the cause. And highly genetic and phenotypic heterogeneity make this kind of disease very hard to be diagnosed with conventional PCR and Sanger sequencing. So this is where whole exome sequencing comes in, as it is a highly effective diagnostic tool to help in clinical diagnosis of heterogeneous disease like this in preventing that. That's all. Thank you.
Thank you. So any questions?
Just one time.
Just one time. Great.
So the technique is not that hard. There is no margins.
Oh,
She should have marks on that.
yeah. So in this case, both mother and father has the stop mutation.
No.
Carry
but because they have the normal gene, so they are living normally.
So it needs both copies to show the phenotype.
Because this one is a type 4A. 4A is a recessive type. So they need both copies to be inherited. So that it is also tested on the younger brother. Actually, the younger brother is recessive, carrying the gene.
So the only comment I have is that you use the term infected incorrectly, because infected means that
Carrying the gene.
they are not infected, but accidentally
Inherited, yeah.
Other than that, yeah, it's very nice presentation. I think most of us can understand very clearly, right? Yeah. Okay. Yeah, it's great. So let's go to the next.
Thank you.
And if you are done, you can choose to like the next.
I'm not ready. No, I know. I want to after finish. Hopefully.
Not here. I think you.
This one.
Okay, wait. To automatically see timer. Wait.
so long.
Maybe you just move the left a bit.是哪个 share 啊？
你是哪个？
我不是没有的，我直接插进去就有了。是 no
Windows
window
P.
screen. There's no screen button. That's why I don't know where's the
I think I have Windows and P.
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 Oh, okay.
Just duplicate.
Stop duplicate.
Windows 加 P 是啥？
就是来看，我觉得呈现这样。你来看这个这个可以选择你的 display 的屏幕啊，
对对对。
一点。哎，也是这个。
Hello everyone, I'm Sana. Yeah, I think my kids are sick. Like unfortunately, like I found two of the cases like they used the next generation sequencing or bioinformatics analysis in the way for confirming their result. So today I will talk about bioinformatics used in clinical investigation of probable diagnosis. And in my case, it's going to be about Listeria monocytogenesis infectious periortitis, a case report from the infectious disease standpoint. As we know, like from the infectious disease, we have a long way of diagnosis we have to follow up until we get the result of the investigation. Firstly, what's otitis? Otitis or periortitis is come from aortic inflammatory disease that the inflammatory of the wall of the aorta and characterized by infiltration of the arterial layer by the inflammatory cells and collagen and this inflammatory cells and responses to the walls of the aorta. It's gonna cause the arterial disruption of the arterial structure. This is a serious case, but it's rarely happen. One of the causes, like they call it by coming from the immunological cases or by the surgical cases and causes, sorry. And the last one, that 1% is going to be due to the after surgical or invasive surgical procedure after done for the aneurysm, abdominal aneurysm by pathogenic bacteria. It happened. So clinical manifestation of this case, dual abdominal pain, fever, fatigue, and weight loss, and also the histological slides show like thick febrile inflammatory infiltrate in lymphocyte and collagen. Due to the diverse clinical picture of this otitis or periortitis, the surgeon they are confused about how to do the investigation or diagnosing the periortitis. Is it belong to the cause of the inflammatory response or causing by the pathogenic? So in that case, like it's really hardly it's gonna be diagnosed correctly belong to which causative agent. Listeria monocytogenes infectious periortitis. As I mentioned, periortitis or otitis coming from two parts, the infectious one and infectious one. The case, in my case, like this one, infectious one, is coming from the bacteria. So everyone can get deadly illness from Listeria monocytogenesis. Listeria monocytogenesis, as you know, it's a fastidious bacteria that we cannot isolate it that much easily in the short period. We have to wait, and the progression of the disease is gonna cause the patient facing a lot of the difficulty. Listeria monocytogenesis, infected periortitis. It's uncommon, but it's deadly. Deadly aortic repair complication. After there's like we have aortic aneurysm, like expansion of the wall of the aorta, they will insert the endografts, prosthesis. Prosthesis help with the aorta wall to manage the pressure of the blood that's coming and going from the aorta. So the complication, it's rare, but it's deadly. Why? Because 25% till 100%, if the patient didn't get the real diagnosis, it will be died by that infection, because this infection, periodontitis, doesn't give any sign for investigation. This science is coming from this case, because this is the only case that they found it, the Listeria monocytogenesis causing the periodontitis. But Listeria monocytogenes causing the infection to otitis, like 7 cases in the literature, but the only one that they found it, this one's from UK. So the signs from my case, it's severe stomach pain radiating to the back and increases CRP, and also from the histological slide, they found it like fat infiltration. It's just the similar sign as inflammatory or deposition of the LDL of the aorta walls. The case here is like we have 76 years old man with a history of chronic obstructive pulmonary disease, both grade 4 and also type 2 diabetes. All that thing, it's coming from like he was admitted for abdominal pain radiating to the back for 10 days, and the pain, it was punctuated by brief episode of diarrhea. This diarrhea, it's made them to think about the digestive tract infection from UK. But the history of the patient, before 9 months, the patient was undergone the surgical things for the undergone endovascular treatment for abdominal aortic aneurysm. So in that point, they thought about to do the CT scan for that patient to find out if there is something like some periodontitis or infection in the wall of the aorta that happened after the injury. This is the profile that they started to investigate in this case. The first one, they did blood test, CRP, and renal function test, liver function test, everything, it wasn't negative. Even for the real-time PCR in the blood, it was negative. There wasn't any signs of the infection. So even they did the specific diagnostic test for this periodontitis or aortitis, they didn't find like there is any signs, there is no elevation of this IgG4. So only from the CT scan and the special CT scan for the artery wall, they found like increased infiltration of fatty tissue around the aortic endoprosthesis, the device they put it inside the aorta wall. Increase of the aneurysm sac expansion, and the most important one, they found it hypermetabolic mass. There is a mass in the wall of the aorta, suggesting that this mass is abscess. From this one, they decided, with many specialists in that field, they decided to do the surgical revision. For what? For just taking out this or checking what's going on inside that process, that they put inside the aorta wall. So in the surgical revision, they found that infrarenal periaortic abscess was noted. So they found an abscess inside the aortic part. So they just like biopsies, they collected and take it out this abscess, and they investigate in that case. So after they went for histopathological examination, no microorganisms found it, and also there wasn't any sign for only the inflammation response. There is no any microorganisms. And then they did the culturing depended, or the way for the culturing in the labs. They found it like after 10 days of the culturing, they found it there is Listeria monocytogenesis after 10 days. So in that case, like they think about that thing. So after 10 days, the patient, even they gave it the treatment to get right from the infection, but then they thought about that thing. This is a real case, like a novel case for them to do the genome sequencing or the cultural independent broad range PCR and bioinformatic analysis, only for confirming the result and also giving the instruction for further cases if it's come to do that way. First way, like they did next generation sequencing by LNS system. The first one, they did the DNA extraction. The DNA extraction was started by the deep microbiome pre-click because they wanted to have the the considerable concentration DNA from bacteria and human, and then after that preparing 1 nanogram of DNA because they needed for my metagenomic library construction. Metagenomic library were prepared from 1 nanogram DNA that like it's contain bacterial and human. As we know, metagenome it means like there is a different kind of DNA, different DNA DNA coming from different cells inside the sample. But the kit they said Nextra XT DNA sample preparation kit according to Illumina system. After the preparation construction, they ran the sequencing, metagenomic sequencing. The library were sequenced, and Illumina made MI sequence instrument they use it, and this is the kit that they use it for. Okay, after the sequencing, they did the bioinformatic analysis from the sample by trimming the rates, the short rate after the Illumina sequencing. They used the Trimomatic package for cleaning and trimming the rates coming from the sequence reader. And after the cleaning and trimming the rate, there were like 3 million, about 3 million of the genes being sequenced. The one that they focused on it was only bacteria. The bacterial one that sequenced 66,000 rates, and this one, by the help of the Kraken bioinformatic tool, they found this bacterial sequence. It's belong 96% belong to the Listeria genus, only the genus one. So all that clean rates after filter it out and label it by the Kraken, label it with the each genus because we have human and different kind of the contamination and the real causative agents. After the Kraken, and they did the alignment and mapping for the only the sequence of the bacteria because they wanted to finding the causative agent. The rates of classified Kraken were retrieved and mapped to complement genomic sequencing. This one they used USearch instead of the BLASTIM. This one it's a search over the search and also clustering algorithm that often ordered magnitude faster than BLASTIM. They did the BLASTIM by USearch, and they found that 96% the same Listeria. From the Listeria, they went to 96% of the Listeria monocytosis. So after that, they showed by the Viewquay, it's a Java application for reviewing, examining prokaryotic genome in a circular and linear context. After they found yes, this is Listeria monocytogenesis, they went for phylogenetic analysis. They found it this Listeria monocytogenesis belong to the strains 6179 in the reference in the NCBI. So this one, it's a computational tool for profiling the composition of microbial communities and from metagenomic shotgun sequencing from the species level. Based on rate and mapping against 1 million marker from the more than 7,500 species, they found this one 99% it's Listeria and 96% is belong to the monocytogenesis species. Okay. After they did that thing, already they know this is Listeria monocytogenesis. But what they wanted to show, what they wanted to do for saving or submitting to the literature, they said this one is the first one, Listeria monocytogenesis causing the periodontitis in the abdominal part. But the second thing, they said, the PCR next generation sequencing, it will be. Oh, sorry, this one is discussion. Okay. They found that everything, it's Listeria monocytogenesis. To treat the patient, they just give the patient intravenous amoxicillin for 6 months, and the patient gets rid of that infection without any further of the complication of its condition. Even though they knew this one, it was the same result, but the same result was employed only, they employed this one for evaluating the effectiveness of using next generation sequencing in the microbiological lab, instead of without doing any cultures. And also there is another they wanted to show that in that result, because sometimes if the patient had the prophylaxis and had the antibiotic, there won't be any positive result of the culture even from the abscess one. So in that case, like culture-independent approach may diagnose microbiology things when antimicrobial are experimentally employed and culture are negative. Sometimes we can get the false negative result. As in this case, it was false negative till they reached to the end of the abscess real sample. But sometimes maybe if the patient got the antibiotic, they couldn't find it with the cultural one. So the second option or the first option, if they can like offer for that thing, they can only do the next generation sequencing, and that's it, because like there's phylogenetic tree, and we can do the classing thing to finding out about the positive agent. This is my reference, this is my main reference, and this is all the main support reference, like help me through. Thank you so much. Question?
Any question?
It's a kind of like why they did it. Why they did only they did it for showing that the effectiveness of the next generation and bioinformatic tools in the real labs, not like in the like because I read a lot of the article. It was all research. Okay, in the research lab, it's okay, they can use it. But what about like in the labs? So in the lab, in this case, like they couldn't just do the only next generation sequencing and giving the result.
So,
Yeah.
so in this research, they are doing the next generation sequencing for confirmation or for diagnosis.
They did it for confirmation, and they wanted, because this is really rarely case it happened, and if it happened, it's deadly. So they just saw an opportunity to show that thing. If like if we face it the same situation, to do directly next generation sequencing without waiting for 10 days or 1 month.
How long did it took for them to?
Next generation sequencing.
Yeah.
They didn't show that information. This was my question about that research. Like I wanted to go do it by myself, but there wasn't enough time.
In this case, she mentioned that they go through the bioinformatics, so they don't wait for the patient to stop the antibiotic and then they will do the culture, because the antibiotic is affecting the culture results.
Yeah.
So they skip this all, because before they do the culture, they should ask the patient to stop the antibiotic.
Yes. From this part, from this level, they gave the patient, after they found the same monocytogenicity, they give the patient, it's because they did antimicrobial susceptibility testing for like because the patient was in the like it was a sensitive situation, so they give the antibiotic to the patient, but they had the sample without antibiotic. So in that case, like they said, they thought about that thing, and they concluded about their research about that thing. What if the patient used the antibiotic for Listeria monocytogenesis specifically, so they couldn't know like the causative agent, it was Listeria monocytogenesis. And this one, Listeria monocytogenesis, it's fastidious bacteria. You cannot isolate it that much easily and in short period. Because most commonly, like this one is going to be 17 case of the Listeria monocytogenesis infection related to aorta. But all belong to aortitis. This is the only case like belong to the periortitis. They found it. So from the point they found that there's differently, they just take the opportunity to do this something and proving that if the patient had the antibiotics, so they couldn't have the Listeria monocytogenesis. They did that one just like from this part after they get the sample from the patient. So from like only by using some of the bioinformatic analyzing computational one, only they need next generation sequencing, and then analyzing by the computational, they can give the result to the patient, and the patient will go to under the treatments and everything is done for patient.
Okay.
Yeah, but this is all belong to the metagenomic analysis. As we know, like metagenomic analysis mostly use it for the environmental microbiome samples.
So the patient has some kind of transplant or
This is
treatment.
yeah.
At the beginning you mentioned.
Yeah. This is one of the causes, because the patient already, yes, because already the patient is old age one, and also he had coped the chronic obstructive pulmonary disease at the fourth level, and also type 2 diabetes. Yeah.
Stent.
Yeah. This one.
Yeah, the stent.
Treatment. Yeah. Because they know the history of the patient, and they went through because there was like radiologists and surgeons and everyone, microbiologists, immunologists, they just gathered and they decided about to do the revision of the surgical thing. Why? Because they knew like what they did, and the patient history is there. There is a lot of the cases like lead to the infection. Otherwise, if the patient didn't have all that history, just go to the patient and they found this in his CT scan, so they will go for the inflammatory reaction, like belong to the immune system, not from pathogenic way. Yes. So like in this literature, they found that like there is infectious one and infectious one. The non-infectious one, you can do anything with it, it's okay. But infectious one, so it's hard to find out. Even the Listeria monocytogenesis is like it's it was inside the collapse of the abscess. It wasn't relating to the like blood circulation.
Thank you. But the only thing is that all of you should have a slide number. So, for example, if you want to refer to slides,
It's 15.
then we can tell
I
you
forget
no problem. So just 6 at a time. So you can select the next speaker. If you want. Thank you.
Just like you will just explain the thing that's in your mind.
Next. Yeah.
Yes.
Go ahead.
Yeah. It was really complicated. I didn't my chance to do that. I found it everything. It was like compared to this.
You can. No.
Topic is AI innovation, certainly focus on the interoperative classification of tumor based on mutual pathways methylation sequencing. Its article published 2 months ago. Yes, introduction. The central nervous system tumors present one of the most dense cancer types, particularly among children. And primary treatment includes neurosurgical resection of the tumor, in which a dedicated balance must be struck between maximizing the extent of resection and minimize the risk of neurological damage and comorbidity. The most common first-line treatment for central nervous system tumor is neurosurgical resection of the tumor. An important factor for determining whether the risk of more aggressive resection is acceptable is the tumor type. But however, in CNS tumors in adults, the extent of resection matters. Gross total resection has been reported to offer survival benefits for isocitrated IDH1 have a globosarcoma of the receptor TRP1 and TRK2 subtype, but not for the mesenchymal subtype. Summary: in some cases of CNS tumors, remove whole tumor is better, but in some cases we can just remove maybe 80% the tumor for survival benefits. And the neurosurgical strategy depends on species and reliable diagnosis of the tumor. Okay, next. Traditional, we use the 3 ways to clinical diagnosis. Use the machine learning to analyze the altered genome-wide DNA methylation patterns. And And we can also use the preoperative image, CT or MRI, or the intraoperative diagnosis, the frozen tumor sections. Use the machine learning, it can be accurate and assign to spatial CNS subtype, but it will spend several days. And use the imaging ways, it always results in a clear, it's not always resulting in a clear diagnosis, and some patients require a second surgery. So they train stringent model for CNS tumor classification and retrospectively apply them on separate neural sequence data. So it can, the model was able to correctly classify the vast majority of sample, 45 out of 50, based on data relevant to 20 to 40 minutes of sequencing, in line with the 90-minute time windows between obtaining tissue in the operation room and diagnosis. So they use the nanopore DNA sequencing. It's a low-cost, small ball factor and instant data available. This is the picture of the machine. So it's very small. And the staging they identify is a neural network classifier, like the patient analysis and optimally tend to deal with the sparse data. That means it use a lot of data to train and validation convergent neural network period to surgery. And next picture is the neural protocol. So one protein will unzip the DNA helix into 2 strands, and the second protein creates a pore in the membranes and hold an adapter molecule. And when a flow of ions through the flow contain a current, each base broke the flow to a different degree along the current. So we can detect the we can analyze the current difference, so we know the sequencing of the DNA. And the adapter molecule is to hold the DNA have enough time to analysis. Next, the methods. They focus on the chosen CNS tumors and consider some situation in actually can happen. So, first, the neural network training wheel simulation. They actually with a turnaround time of 90 minutes, only very limited nanopore sequencing data can be generated, and so they use so they develop a simulator strategy like generate realistic training data from array-based methylation profile, and they consider some situation like the sample purifier very sensitive, because when we actually surgery, we can take the whole tumor tissues. They may have include some normal cells. So and they have other problem like the copy number variation. Some sample have difficult to specify as a astrocytoma or oligodendrogloma based on the methylation profile and historical. In such case, the YP and ITQ deletion offers strong supporting evidence for the identify. So in the copy number variation, there are some chromosomes we miss some genomes, and there are 2 strategies. The location-specific models, they can focus on the specific tumor, use the specific model, and adaptive sampling. Because when use the nanopore sequencing, we can immediately to take a result. So if we run 400 base, we can know this tumor is the target, it can be recognized or not. Then the result. So first, they very stimulated spread nanopore from this of the nano arrow data for model training. First, they filter the results. Use the 0.6. The number is higher than 0.6 is 1 and less than 0.6 is the 0. Because it's the limited of the technology, it may lead to a underestimation of the true methylation level of at least size. And they use the array to make a lot of the show sequence for training. So they use the two folds to training and one to a validation and final fold to testing. Okay, the next. So the each box means there are seven boxes. That means the first 10 meanings, 20 meanings, 50 meanings. So almost the subtitle can recognize in the 15 meanings actually. This is the sample purified digest sensitive. The different sample including cancer cells and normal cells. And the zero means no cancer cells. The yellow culture means normal cells. And in the correct results, green, the darker means the darker green means the hydrogen classifier has a high confidence to control the cell type, and the lighter green is the Heidelberg classifier unsure part, but it actually is correct. The Heidelberg classifier is a tool it also a tool to identify the cell type, but in these results, the model labeled is better than the Heidelberg classifier. Okay. So they use the last result, they use the simulation, and this result they use the nanopore sequence sample. And I think it's a similar result because they are also can recognize the tumor subtypes in maybe half hours. Okay, and then they use to demonstrate the clinical festival of the certain gene interoperative sequence context. They performed the protocol during 25 surgeons at 2 different hospitals in New Zealand. Certain gene was able to classify 72% of the tumors, and subclass level with almost 45 minutes of sequencing. And rapid molecular classification will have been of substantial added value. For example, in two cases, the tumor class was unknown prior to surgery. In intraoperative frozen section diagnosis suggested an impandenoma in both cases, and indicating a radical recession as the best cause of the section. And shortly afterwards, Sertinger achieved a high confidence for ependynomal subtypes, and the both classification will later components use a methylation error and the Heidelberg classifier. So actually, the sequencing and get results is 30 minutes. The most time is used in the sample collection, DNA isolation, and preparation. It's a 90 minutes the window in the surgery. It's the discussion. Because owing to the short sequencing time, only very sparse data are available, but in the future, more and more narrow sequencing reference samples have been produced. The model can analyze rapidly and higher count rates. And a potential limitation of the Sertinger approach is the required most of the tissues. And although there is a sensible extract the sufficient DNA from small sample, including a biopsy, the biopsy is the putting a long needle inside your body and pick up some tissues. Okay, and to rapidly obtain a high concentration of DNA, the DNA extraction protocol optimized to extract DNA from fresh tissue sample in 70 to 20 minutes. They make it faster, and it's necessary to find out ways to optimize detection ability of Sertinger model in the misclassification and difficult to diagnose the case. And another questions I always confuse is it's allowed when we open patient's hand and waiting computer results the 90 minutes, right? And continue the surgery. But I asked a friend, it's a daughter, so he said it's okay, we can wait 90 minutes and then continue the surgery. Okay. Conclusion is the future graph methylation sequencing has enormous potential in other domains such as regular post-operative diagnostic, global to turnaround times, and antibody use in peripheral and low-income institutions. Okay, that's all. Thank you.
Thank you. Any questions?
It's really, you can wait for 90 minutes?
Yeah, because there are a lot of diagnosis tools. Maybe you need at least, I think, 12 hours.
Yes.
Because it's important to the patient's survive. Yeah, because you can identify the tumor subtype. It's very important for you.
Because in our hospital, 45 minutes is the maximum. Yeah, we have to, I mean, within that, have to give the result to the doctor. So the site is quite surprising, you can do it for 1 hour and 30 minutes, right?
So after knowing the tumor subtype, then what's the action by the surgeon?
They can decide we should remove the whole whole tumor tissue or just maybe half, or it depends on
There is a grantee from depends on whether tumor is fast growing or slow.
Maybe certain margin are involved.
Is it because some tumors are very slow? Then
I think it's not depend on the size. It depends on the subtype. Some subtypes it may be huge, but you can remove whole of the tumor. Yeah. Because the brain is no, it's unlike the because it's something like the liver. You can cut the liver, but you can cut half of your brain. Yeah. Okay. Thank you.
Like what I say, English.
Yeah.
We can take a short break.
Brain cancer first.
So I think in the future you can summarize.
Breast cancer, you can brain cancer, you can
Maybe too much or yeah.
Mine is totally same with year. My case is the same as year.
Doctor, my topic is not presenting on case study.
We shall see.
How? Don't give me low class then. Because I didn't.
No, like, it was so hard to find a case. Okay. The one that found is like you. Yeah.
My topic is to talk about future prospect of developing some algorithms that integrate, which involve integration of multiple of data.
Yeah. Okay.
I don't know why, because it's not related to like some cases because basically it's just finding development finding.
But you can apply to some
Should I say the presentation?
use, right? Can be applied.
I mean in the experiment,
Yeah, sure, maybe.
Yeah.
No, the 肠胃道。
肠胃道。
No, like the burn size and the
How this cycle? What's how we now?
锅巴。对，它很锅。
锅巴就是肠胃道那个，高用 five spices power。
Spices power.
I don't know how I got it and a little bit of okay.
You can sit down first a while with me.
I'm very simple.
No, no, for reading in USM. No, So we have one more quiz on this course. So when is the good time?
When is a good time? Yes.
So it's an online quiz, but you can cheat. Yeah. No, just a short answer. Yeah.
Think between after Christmas camp.
Five. Yeah, that's the 5 topics.
Chinese New Year lah. Then you all get 0 marks already.
First or second week of January. Yeah, but later on might have more assignments. Oh. You can do it at home. Don't have to come here. Fix the time then.
No, we have to come here.
Those who want.
Open book ah. Yes. Open discussion.
So next Friday. Do you want at night or I don't know because some of you guys are working, right? Saturday only can do it. I just set the time. As long as everybody agrees at the time. And yeah.
Next Saturday.
She's working.
It's not after New Year.
No, because
I'm working Saturday.
more assignments to join. Because you said more assignments and
Sunday.
better to finish.
Christmas.
You can do it very fast. You can finish with the instructions.
Saturday cannot, Friday cannot, Sunday cannot, when can? Friday.
Yeah. What time? Evening. 8 o'clock.
加到那个地方，跟他请假，对呀，这节课不是取消了吗？然后昨天不是加到礼拜5，他为什么不能加回去礼拜3？他不是礼拜2不行，礼拜3。礼拜3不能他不在这里，他在朋友。他是礼拜5，他到礼拜3。
22nd. Job is decided.
22nd ah, okay?
8 p.m.
Okay.
Yeah. Just 1 hour, but you can finish. So you have a quiz for this part. The last assessment of
That one doctor do what assignment when
next Friday at 8 p.m.
Friday, 8 p.m. Oh, what's up?
Next week I will very full.
Yeah, all the topics. Well, one topic, one question. Because there are 5 topics in 5 minutes. That's when we decide first, then let me know. Everybody decide, let me know. What?
His subjects. And then we were like,
Quiz for the whole day.
whole day. So everyone please go in.
Yeah, sure. The whole day, right?
玩死人
很好吃，你这个会一直想要再吃掉。
C
Good time.
29.
So the time will be open for 24 hours. Then.
I think everyone is
So which day is
Sun. 29.
just the day before New Year?
No, 31st.
At 8? Is that?
No, it will be the whole day. And that time is just 1 hour. On for 24 hours. After midnight.
看你好，你的慢讲可能泡汤嘞，是你的慢讲，来去吗？
下礼拜的会。
奖励自己做不到那个期待，大概差不多都这个他30，这个他今年学工资要没有毕业，他就是。As the patient levels come back to normal, and for only outside of 6 years old, slow down. Yes. Down, still many problems. 哦，对对对。我妈不是没有真的是哦，这个是我妈。那我可以从英国做水可以吃。
So when
If they see satisfaction,
Yes.
if they
Yes,
see,
tell them.
tell them.
tagging you.
Oh, yes, I do.
Please don't exceed.
So communities and everyone. Today I'm going to present to you the applications of the bioinformatics in the clinical diagnostic. Case study on the diagnostic design in a patient with HIV infection. Okay, before we dive into the case study, let me go through the basic information of the diagnostic design again. Okay, previously known as diagnostic parallel pneumonia and also initially classified as protozoans. However, after the careful phylogenetic analysis, it is now reclassified as in a fungal family. This pneumocystis jirovecin pneumonia is mostly affected the immunocompromised patients and sometimes can be severely life-threatening. For those patients who have a cancer, organ transplant, HIV, congenital immunodeficiency, and who underwent the immunosuppressive therapies and medications, they are prone to infected by this pneumonia. The primary route of the transmission is through airborne. Okay, the pathogenesis of the pneumocystis jirovecii is when they through the inhalations, when they enter the lungs, okay, the trophozoites will attach to the alveolar type 1 pneumocyte by the pneumonia is like filament projections from the trophozoites. Then, while in the lungs, the pneumocystis has biophases life cycle featuring they will undergo the sexual and asexual reproductions, replication inside. So, in order to save my time, so maybe later we can come back for this cycle. So, adherence of the pneumocystis to alveoli is not the only cause of diffuse damage, but rather is our own host inflammatory response cause the significant lung injury and impair the gas exchange, leading to the hypoxia and possibly the respiratory failure. Okay, before that, let me go through the background of the case study. The 22 years old Chinese male from the Zhejiang Hospital and complaining of the persistent day intermittent fever with a temperature of up to 38.7 degrees Celsius and 3 days of progressively dyspnea and red hot. He was given the moxifloxacin at the hospital. However, he developed chest thrust after using moxifloxacin, so after that he stopped and given another drugs and also hydrochloride. Due to the history of the male sexual partner and weight loss, so the hospital decided to do the HIV test for this patient, and the result come back positive, HIV positive.
Thank you.
Okay, so from the day 1 to day 4, okay, the first day, he admitted to the first affiliated hospital, School of Medicine, Zhejiang University, and then the symptom still persist, still had a fever, dyspnea, fatigue, cough with a small amount of sputum, and some pathologia and dyspnea during activity. So the serology biochemistry index, for example, like LDH, show higher than the normal range. The 1,3 beta D globin test show positive, the Rhabdomyinum test and the Cryptococcus capsula antigen test were negative. Then the nucleic acid amplification test of this COVID, Mycoplasma, the influenza A and B were negative. The Epstein-Barr virus test, MDB-PCR, GMS staining, and the culture of bacteria and fungi from the urine and sputum blood specimen show negative. And however, the CMV, the cytomegalovirus test show positive. And then the CT scan here, you can see that from the image B, there is ground glass effect shown on the lateral lungs. And on the image A, you can see there's a thin wall of cavity lesions as shown in the as the arrow. Then on the day 5, in order to verify the pathogen-induced pneumonia, so patients underwent the bronchoscopy to collect the bronchoalveolar lavage fluid specimen. This fluid was sent to the laboratory for conventional test and also the NGS test. So the conventional test, RAM stain, AFB stain, G test, GM test, and anti-PCR that we mentioned just now, all show negative. On the day 7, means after 2 days, they collect the fluid specimen. Then the results show that the pneumocystic jirovecii and the cytomegalovirus were found in the fluid. Then the other microorganism also detected, but because of it's not regarded, so that's why we ignore it here. So the diagnosis of this multi-pathogenic and pneumonia due to the pneumocystic jirovecii and the cytomegalovirus. So the treatment, after they can confirm this diagnosis, so they can start the treatment with the ganciclovir, intravenous ganciclovir. So on the day 7 to day 14, after admitted, the symptom is disappeared already after the treatment. Then the general condition in normal laboratory finding is improved. So the chest scan shown here, is like already has been significantly absorbed, but except there's some faint patchy opacity in the lateral segment, and the thin wall cavity lesions still can see, but it's already reduced in size. So after that, the patient is discharged and start the antiretroviral therapy for the HIV. Then after 2 months, it's come back and follow up and check the patient. Everything is okay, appear healthy with no symptom, and the CT scans now have no any animal finding, except there is a faint patchy opacity in the left lateral segment. Okay, so that's the case study that I mentioned earlier. Actually didn't go into details of the bioinformatics workflow, but therefore I do some study on other research and other literatures. Okay, found out that this is a general bioinformatics workflow that we will use to identify the insider single or multiple pathogenic pathogens. Okay, so first, for the bioinformatics workflow, we will have 2 components. One is a wet lab pipeline, one is a dry lab pipeline. The wet lab pipeline, okay, is including the when we collect the specimens. Now after that, it undergoes the clinical process, including the nucleic acid extractions. Then after that, the sequencing library preparations, and then the sequencing adapter attachments, and after that, we go to sequencing. So next, we will go to the dry lab pipeline. The first one is the base calling. Okay, base calling is where it's a computational process where the intensity of the signal light will be captured, and then when the nucleotide and the nucleobase pass through the nanopore. So for that, the DNA sequencing data will be obtained. So after that, the target sequencing reads will be go to the preprocessing. The preprocessing is to like exclude out remove out all the like short read, for example, the length less than 5 base pair, and then remove the low quality, remove the low complexity regions from. It will be removed. This is the preprocessing. So after that, the preprocessing, the sequence read will
aligned to our human database, so that in order to remove the human reads, because most of the sample from the, is that from the host is human, so that's how we want to remove it out. Then after that, the preprocess and also the non-human reads, sequence read will be aligned to the pathogens database, and also assign the taxonomy classifications for each sequence read. And then we use the organizational and the statistical analysis of the result to identify the microorganism. So this is the biofilm workflow for the general biofilm workflow for detections of the pathogens. So the discussion for this pneumocystic cerevisiae pneumonia is an opportunistic pathogen and widely found in the lung tissues of animals. When body immunity is low, it can lead to the pathogenic fungal infection in the lung. So if failure to receive timely treatment and appropriate medication will lead to the progression of a severe respiratory failure and septic shock. The case study identified multiple pathogens. The first is pneumocystic cerevisiae, around the 1 million reads, and the cytomegalovirus approximately 4,000 reads. So the diagnosis of a multi-pathogenic pneumonia involving both pathogens was made through the NGS and bioinformatic analysis of the fluid. So despite obtaining the negative result from multiple conventional diagnostic tests that I mentioned just now, so study also reported that twofold higher mortality at 3 months in patients with pneumocystic cerevisiae pneumonia who also have a cytomegalovirus culture. So the gold standard, to detect the pneumocystic cerevisiae is through the Gomori, Lockhart, methylamine, silver nitrate staining. But unfortunately, in just now the case study is shown negative. But fortunately, is the NGS able to detect it. So the conclusions, especially beneficial. NGS especially beneficial to the HIV-infected patient who develop pneumonia and are more likely to be infected by multiple pathogens. So if there is a lack of the infectious disease diagnosis or delay in diagnosis of infectious disease, it can become life-threatening. Then the NGS is an unbiased approach that is able to detect and construct genomic sequence on nearly and all pathogens, including virus, bacteria, fungal, and parasite. With this approach, it can accelerate diagnosis and lead to the healthcare cost reduce and improve patient outcome. I think this is the ultimate outcome that we want. 10 minutes. Okay.
Oh, you got the NGF from the biopsy also? Or is it like from the blood?
From the bronchial alveolar lavage fluid, we threw the bronchoscopy.
Oh, so it's some kind of biopsy also. Consider as a biopsy? Or different?
Cytology.
Cytology. So if we look at the CT scan, the one with
the first one.
The second, the last one.
Last one.
The last one.
From this extent.
Yeah, I think this one, right? Oh, okay. So after they do the treatment, but still there's some.
Infections, but everything is resumed to normal.
Yeah.
Any symptoms.
But still there is something there.
Yes, it's like
not
a
clear.
side effect. Maybe like after COVID, there we get some side effect.
Okay. So is that the one that is the infection?
This one,
Yeah.
it's no longer, it's like the lesion, it's like maybe after
Oh,
infection, so there's maybe left some side effect.
Okay. Okay.
So maybe while doing some very vigorous activity,
Yeah.
What therapy they use?
Antifungal.
but we need to really identify the exact, which fungal, so they can give the proper exact treatment.
Thank you.
Thank you. Coming back.
Yeah, the bring that up. That.
Beautiful one.
So very
So I'd like to give a heads up. It's actually the same case study as Yuan, but maybe I will clarify more on the testing and analysis part. So will give us a clearer idea of what happened behind the screen. My topic today, muscle atrophy diagnosis by bioinformatics analysis on whole exome sequencing. So muscle atrophy is a progressive condition that causes loss of muscle mass and weakness in different entities. It's a heterogeneous disorder which causes defect in peripheral nervous system, central nervous system, and also neurotrophic deficiency. So many genetic diseases have multiple causal genes. Overlapping muscle atrophy phenotypes lead to insufficient disease knowledge and diagnostic errors. If you can remember from Yuan's presentation just now, she did mention that even cancer patients can have muscle atrophies. So we will not exactly know this cause, this specific person who has this symptom. Is it from a genetic disorder or cancer? So it's quite difficult to figure it out. Because as you can see, even mutation in the same gene can lead to different forms of phenotypic abnormalities. For example, when you have a mutation in lamin AC, LMNA gene, this can result in maybe Emery-Dreifuss muscular dystrophy, dilated cardiomyopathy, or shallow Merry-tooth disease, or spinal muscular atrophy. So this complicated genetic and phenotypic heterogeneity possess obstacle to rapid and accurate diagnosis. So eventually, if the patient will go through, we call it diagnostic odyssey, because they have to go through a lot of testing, it consumes a lot of time and money. So there comes next generation sequencing. So this is revolutionizing the genetic diagnosis because the massively parallel sequencing is much more efficient and affordable compared to whole genome sequencing, as we are more knowledgeable on that, right? Whole genome sequencing. This whole exome sequencing, it only analyzes the coding regions. So it's comprised of 2% of human genome, but this 2% harbors more than 80% known Mendelian disease associated variants. Why I say Mendelian disease? Remember Mendel's experiment of inheritance using the pig plant? So that is the fundamental for Mendelian disease. So WES is rapid, unbiased, cost-effective sequencing strategy for elucidating genetic variants of human disease. WES uses a family trial consisting patient and healthy parents. Trial exome sequencing, that means use the healthy parents and also the patient, so that we can eliminate the family variant. So now let's look at the case study. This is a case study of a Chinese family from Guangdong province. This case was reported by Jiang et al. In 2021 and it was published in BMC Neurology. As you can see from the pedigree diagram, we have a healthy parents. We have 3 kids. The eldest son was healthy. The second one, daughter was, and the third son had this muscle atrophy. So we're going to look more in detail what happened to the second daughter, which was the patient. So the patient was a 29-year-old female. She presented with severe myotrophy and distal skeletal deformity. At age 5, she had stumbling gait. I mean, she could not walk properly, disoriented. And then she developed an ankle contraction and foot bending. So acupuncture treatment was done. I believe maybe it was quite popular at her region to have this traditional treatment, but it was not effective. So at age 10, a surgery was done, but the condition did not get better because her feet became weak. She did not have exercise. So the extremity muscle wasting, her muscle size decreased, and it got worse. Finally, she was wheelchair dependent. So her recent physical examination was she had bilateral muscle weakness, atrophy of both her lower and upper limbs, distal deformities of severe valgus ankles and wrists. Her ankles were distorted, and contracted feet and palms. Was like practically inclining inwards. So this is the diagram, but she didn't have any other apparent symptoms. She was fine. She was speaking, hearing, and intelligence was fine. But the similar symptoms was found on the younger brother, remember? So this is the picture of the patient. She was wheelchair bounded and she had this distal skeletal deformity. The picture above shows her severe weakness and atrophy of lower arm muscle, valgus wrist, and contracted arms. The lower picture there, weakness and atrophy of lower leg muscles, a bit dropped. So this is the typical symptoms of this disorder. So this disorder has a lot of other genetic, it's the same phenotypic resemblance to other genetic disorders such as BMP1 for osteogenesis imperfecta type 13, HOXD13 for brachydactyly syndrome, and SAC24D for Cole-Carpenter syndrome too. But all these genetic testing turned out negative for this patient. So now there comes the whole exome sequencing. I would like to briefly tell you what is whole exome sequencing pipeline. As you can see here, remember the trial exome sequencing, it was done on the parents and the patient. So first the sample preparation, usual, DNA extraction, and the DNA was purified. Now we go to the target enrichment. So what is this? Because we want to get the exome part only, right? So we need to get, we have to fragment the DNA, and in order to get exome, we utilize exome capture technology. This technology is actually, after the shearing, the fragmented DNAs are hybridized, where only the exome will hybridize to the microarray. So after the washing and solution step, we will manage to get the exomes. Still later, it will be sequenced and analyzed. So this is the bioinformatic analysis pipeline. The raw reads that we get after the sequencing will be aligned using Burrow-Wheeler aligner tool. So this will align together with the reference human genome. So once the aligned read is obtained, the reading is trimmed and the exoming reads is analyzed using genome analysis toolkit. This will give us the SNVs, meaning single nucleotide variant, and the short insertion or deletion variant. These are the mutated genes. So from this reading, further analysis can be done using KGG sequence, which will filter, and the annotation will be done. Finally, we can get the disease-associated variant. So based on this study, these were the findings. They did the WES, they managed to get the WES data from the parent and the affected patient. Using the trial-based variant calling, the GATK, Genome Analysis Toolkit, they managed to get 1.5 million variants, meaning the mutations. Out of this was 1.2 million single nucleotide variant and 226,000 of the short insertion or deletion. Then, using the KGG sequence, they only focus on the coding or the splicing region, the exomes, and minor allele frequency less than 3%. With this, they managed to get 1,280 rare variants in that patient. They focus on the mode of inheritance because we are looking at the muscle atrophy. So we are looking at the autosomal recessive or compound heterogeneity. They managed to get 22 compound heterogeneity and 5 autosomal recessive variants. So out of these variants, further analysis was done. KGG sequence and bar cuts were used to predict the pathogenicity. Out of these, 5 variants were showed after the analysis. That 5 variants were further analyzed for genotype and phenotype correlation. Out of the 5, we managed to get 3 candidate variants. These were the C218 in GDAT1, C2029 and C1262 in ARHGEF11. So these 3 variants are further validated. How they validate? Using Sanger sequencing. So as we can see here, this is the result from the Sanger sequencing. Remember the 3 variant, we had the GDAT and the 2 from ARHGEF11. If you can focus on the nucleotide here, the first one is the normal control from the Human Genome Project. So first is the second one is the father, mother, elder son, the patient, and the younger brother. If you can see here, this is the normal one, C. The father had a C, the mother had a G. But the elder son had a C, which is a normal, correct? And then the patient had a G, which shows the abnormal one. And then the younger brother had a G, which is also the abnormal one. When we look at the second and third, we can realize that the normal one was C, father had a C, mother had abnormal T, elder son C, the patient had T, but then the brother had C, which is normal. That means it doesn't correlate with the phenotype. And also the same case happened on the third variant as well. So now we can, from this sequencing result, we can conclude that GDAT was the one that was causing the mutation. So finally, they managed to detect is a homogeneous nonsense mutation of ganglioside-induced differentiation associated protein 1, GDAT1, which is the subtype of this disease, Charcot-Marie-Tooth disease, which was not, they were not able to figure out this disease even before this. So it was confirmed by the nerve conduction velocity and electromyography. So what is this Charcot-Marie-Tooth? Charcot-Marie-Tooth comes, the name comes from the three physicians who was the first one who diagnosed this disease, Dr. Jean-Martin Charcot, Dr. Pierre Marie, and Dr. Howard Henry Tooth in 1886. So it is also known as a hereditary motor and sensory neuropathy, HMSN. Inherited peripheral neurological disorder. It causes muscle wasting, weakness, and sensory loss. Two categories is CMT1 and CMT2. CMT1 is actually the demyelination. As you can see from the diagram, we have a myelin sheath covering the axons. So what happens is due to the mutation, the myelin sheath damages, it decreases, or it can be CMT2, axonal, the damage to the axons itself. So now GDAT mutation, GDAT1 mutation have severe phenotype and become wheelchair-bound patient. The patient becomes wheelchair because of this mutation. So the diagnostic examination for this disease is neurological examination. When the patient demonstrates peripheral neuropathy, probably they will suspect for this. And electromyography is to measure the electrical activity of the muscle, and nerve conduction measures the strength and speed of nerve electrical signal. So it's quite general. We couldn't, even if we've done this before, we couldn't even figure out this is the disease because it's the same representation for other many genetic diseases. So this Charcot-Marie-Tooth disorder has no cure, but definitely management can include physical therapy, orthopedic intervention, which include braces or orthopedic surgery, and also assistive device for mobility. As a summary, combination of WES whole exome sequencing with bioinformatic analysis will give us an effective strategy for Mendelian genetic disease. WES is more cost-effective, diagnose genetic disease with atypical clinical manifestation. It's not very common, so using WES targets a whole round of disorder. I mean, we can detect from whole round disorder, this is the specific one that is causing the patient disease. So important role in early diagnosis, prenatal and postnatal care, medication guidance, and also new drug development for hereditary disease. So thanks to WES, at least the patient has now know what is the cause of the disease, and the brother can be treated as early as possible. So my main reference, Zhang et al. These are my supporting references. Thank you.
Any questions?
So this gene, right, is the one that affecting the myelin sheath inner.
Myelin sheath.
Yeah.
CMT1, yeah.
Okay.
Yeah.
So it doesn't. So it's what affects the instead of the.
Because when the myelin sheath is decreasing, now the
signal
neurons,
is able to.
yes, the neurons is affected, it's damaging. So the muscles cannot get the electrical signal, the proper electrical signal. Eventually it decreases.
I still call myotrophy.
Myotrophy, muscle atrophy.
Atrophy.
Yes, muscle atrophy, because
not able to walk. So when you're not able to walk, you will lose your muscle mass.
Sorry, I was thinking about this disease is known as muscle
Sensory dystrophy cannot feel.
effect in your sensory, because there's no, I mean, if you touch something, right, you will have that sensory to and fro. But once your axon is affected, your neuron is affected.
I was thinking about muscle dystrophy, but I was thinking about this is the muscle degeneration. Muscle degeneration, I thought muscle is not.
Yeah, thank you.
Yes. Yeah, thanks. feel like don't know anything. You're like, okay, macam, yeah.
不需要，不需要，但是我是有一个这个论文的，因为我也很方面，因为我这个短期的只是这个礼拜天才做嘛，所以我要把这个就用别的剧本，大家有讲。
哦，台湾是有带
你们呢？你的在哪？你没有吗？没有做到。
做了是，那
Alright, start now? Yeah. Okay, so
So,
really to my long-term colleagues, so Yang Zijian, so today I'm going to present you about something a little bit different than what are you guys presenting today. Yeah, which is not solely focusing on the design itself, but on the bioinformatic tools itself. Help us to
from business part
to generate molecular profile. So, okay, so a prologue is, so with the current blooming of computer technologies, bioinformatics has become increasingly significant in various disciplines, especially clinical practices. System bioinformatics is also a branch of bioinformatics. It's a study of focusing on integrating information across different levels using a bottom-up approach as in system biology, data-driven top-down approach as in bioinformatics. What does it mean is that we are trying to, system bioinformatics is a study of focusing on how to integrate all the data together into creating something, a powerful tools for you to do these three things in clinical practices. Diagnosis, prognosis, analysis is mostly significant here, and also personalized medicine or treatments. Okay, so next. So overview on my presentation today. So the topic of the presentation today is mainly about integration of multi-omics data with scoring-based analysis network as a potential bioinformatic tools for biomarker discovery in biomedical studies. So one of the things that all the case study presented today is more towards like Mendelian disease or very well established cancer studies, a very well established cancer. So, but somehow for some other complex disease, right, they involve a lot of different pathway biochemical reaction, which this gene is at as an upstream. This biological molecule is in upstream, which may affect some, brings about various pathway. So how do we overcome this? Is by web lab is very impossible because we couldn't know what is the other effect, the hidden effect that these biological molecules may cause us, right? So what scientists trying to do today, this is very new because all the journals I found is mostly in 2022 and 2023. So introduction on this, yeah, like I mentioned, traditional analysis of a single omics data often means the complex interplay between different layers of the cells. Hence, integrating the omics data with a scoring system to determine the pathway of the biological molecules in cancer development. Sorry, this is thin and thick. In many disease development is getting more trending across cancer studies, in these studies. So in this presentation, the aim is to review how does these two tools, integrating this omic data together with a scoring system, can lead us to into a biomarker discovery in silico, which means that you don't have to do a wet lab for you to discover those biomarkers. By having the present data and also a powerful tools, machine learning tools, you are able to even predict what is the upcoming novel biomarkers in the cancer. Okay, so just to fresh up your knowledge, what is omics data? So we have a lot of omics. Genomic is the popular one, where you guys study the genes. Transcriptomics is the one after you transcribe. Proteomic is the protein, the product. Metabolomic is the metabolite, yeah, that the protein may cause. And then exomic is like just now they do the whole exome sequencing. That is the study of exome. Called as, some people call it exomics, but I'm not sure what is the appropriate word here, so I just put exomics. I mean, some studies journal use this word as well, but it's not recognized yet. So a scoring system, what can a scoring system be? It can be as fundamental as these three: a sequence similarity scoring, structure similarity scoring, and functional similarity scoring. This is very well established bioinformatics tools that's available for quite a long time already. What does it mean is that we have a lot of these, like let's say it's sequence, right? So we have a lot of these genes with a very similar sequence, but not 100%. We use the scoring method into correlating them by the identification of the sequence. Okay? Structure is related to something to its structures. Functional means its function. This is a very fundamental scoring system that's available for quite some time already. So what are we looking at is a higher level of scoring system in order for us to just by a tips of hand, just plug inside this input and then we can just get the prediction result that we want. Okay, so a more advanced level of scoring system, it might be a network-based scoring, which network-based is you can include a lot of these different parameters, and even a more powerful tool one is the machine learning-based scoring, which you create an algorithm with a present database. The tools, the software you use is able to give you a prediction result, which I'm trying to mention. Yeah. So, introduction. So a method of study. So as for now, there are still no standard method in producing the best result. What I mean is that because there is still no any proof that which combination, because there are different many scoring and different omics. So there's still no result on like which combination of these omics and with the scoring give you the best idea of the biomarker discovery. There is still no consensus among scientists nowadays. It's still in argument. So the aim of this today, as also a future prospect of this, is also want to develop a gold standard tools to do analysis, right? So method of study. There's a lot of method of study by integrating with your omics. Okay, so we have for example, a kernel-based, which involve a lot of these different kernels, and then it's a so the one of the example here is I'll give you SNF, which is similarity network fusion. Later I'll mention it again, and there's many network-based like this. Very, I won't say complicated. It's all of this is about statistical model they created for you to do a scoring system. So this is what you can do. Okay. So overview. Okay, finding examples. All right. So what I find, give you a case study today is something related about cancers. So why cancer is important according to Global-Can 2020. The total cancer riser simulation in 2020 is 48,600 with 29 included motor cases. It's actually quite high in Asia as well, cancer. So cancer is a heterogeneous disease, which means that it is a very complicated disease caused by changes in cell behavior, chondral growth, and genomic mutation, etc. Can be divided into many subsets. But in clinical, when we're talking about cancer, right? How we generally categorize them in prognosis and for treatment study is that one significant approach is categorizing them into genetically similar subgroups and associate this group with clinical outcomes. That's how conventional studies on cancers are done nowadays, which is also used by a lot of papers. So these are some of the popular cancer, not popular, the cancer that infected the most, not infected, how to say that? Common cancer, yes, common cancer. So you have BRCA, which is a breast invasive carcinoma, like the breast cancer one. Colorectal, which is also apparently the second highest cancer in Malaysia. Lung, I think, is the third highest, and kidney, I think, is more overseas. But this later on we'll talk about these four more, because in these findings, what I do is they're focusing on these four types of cancers. Okay, so this is a summary workflow on developing algorithm that can lead to a cancer biomarker discovery. So it's depicted from these two very genius guy. It's yeah. 我们用 better analysis animation. Yeah. So at first, what the parameter they trying to I know it's very hard to digest this. I went through some supper. I suffer a lot also. So one of the two important data set we can use to study cancer is the differential gene expression and also DNA methylation. So gene expression, I think you guys know what does it mean by gene expression. So those oncogenes, tumor suppressing genes, and then DNA methylation is the. You guys understand what's DNA methylation? No. Okay, so when DNA is said to be methylated, it means that that DNA is silenced. It's something that you add something on that, then your transcription will not be able to done. That's methylation, silencing them. Okay, so this two is highly related to cancer because cancer has the ability to control what genes being expressed and what genes should be silenced and what genes should be upregulated by removing the DNA, the methyl group associating with your DNA. Okay, so first of all, what they do is they identify of a differential expression genes and identify this. Later on, this also is a sign of probes because you can't do this. The method to do this is a sign of probes. Then, okay, so after that we identify a common genes, okay, which integrate these two. We found this, which these two is common among these two, because gene expressed is something also related to gene methylated, right? Because we want to suppress the gene also or activate the gene. So identification of common genes. Then we normalize them, and then they will construct a gene express similarity network. This network is, they use, I think in the paper they use 3 algorithms to generate these networks based on different parameters. Okay, the parameter is maybe the modularity, but what you can, the basic concept here is the similarity between them and creating a network. So imagine you have this gene as the first node. So if let's say this gene, the second node is related to the network of that. But different algorithms have different statistical model of this. So you guys can read on that and what model you can have. That can be your sample also. Okay, so you fuse a gene in a similarity network. Then after that, a network based. So this genes is maybe a potential biomarkers already. Yeah, we are talking about this gene because it's a common gene, so it's a potential biomarkers already. So what do we do next is that we want to do so fast network-based clustering for both training and validations data set. Okay, so we find out this gene already, right? So they take the samples from what available in patient training samples and validated samples. Then they correlate the genes they found here and then the genes they use here to identify the common genes in the same modules. And then that will be another different algorithm. I think I should express more, but I think the time is lacking but. So and then they integrate for mutation data. Finally, they go to a survival analysis. So all this after they all get the biomarkers ready, right? So what the process is actually filtering out the previous biomarkers. What I say is the potential biomarkers. So why they do this so much networking is that we want to see if there's any correlation or not. So the non-correlated one will be filtering out. The basic concept of this process is here. So finally, all these genes will go under this survival analysis, which is a statistical model designed by these two further, and to relate this effect of this gene on the survival time of cancer patient, and then lead to a biomarker proposal. Okay, I hope you understand. So this two is the database available for the cancer genome. I mean, yeah, TCGA. I think it's common, right? TCGA databases.
No, maybe for cancer researchers.
Yeah, cancer researcher is a very, very common database. Okay. So in these studies, the common gene modules across different cancers that are strongly associated with prognosis is discovered. So all these genes is actually, so the reason why I mentioned about that 4 genes because the 4 cancer is because all these genes is somehow related to all of them. So we want to see whether all of them have a correlation or not. So this is the result defined. Hypomethylated means that the genes, this is potential genes, biomarkers, they can count in cancer types. Hypomethylated means that it's the genes that's polymethylated. Polymethylated means that it's not methylated, then it's activated. It is a possible oncogenes. And the hypermethylated one, it could be a suppressed gene. So this biomarker across each cancer type is covered, which I rated by the prognosis score, is aligned with the biomarkers discovered by a wet lab. They provide a lot of reference on the wet lab experiments, which is all this as aligned with all these biomarkers that's discovered by wet lab experiments done before. So which is actually quite efficient, if you want to ask me. Okay. So as a concluding mark, integrating multi-omics data into analysis is the future prospect of bioinformatics in coming era. It's anticipated that the combination of these omics will generate a more comprehensive molecular profile either of the disease or of each specific patients. Okay? So it's just not only disease you can do a patient one as well, correlating the patient. This molecular profile is expected to make several highly ambitious objectives, such as the computer-aided diagnosis and prognosis. In identifying disease subtypes, there are still many cancer subtypes we still don't know. So that happens in the rare patient with the rare patient. Okay, the detection of complex molecular patterns associated with disease, the understanding of regulatory process involved in disease pathogenesis, and the prediction of response to drug treatment. All of this actually can be done because actually you are correlating them. So the aim here is that, however, there's still several challenges still remain unsolved. There are several challenges still remain unsolved. So what are the challenges that still remain? Okay, so data heterogeneity. So it has a lot of different data. As I mentioned, they still have no any consensus on which omic is the best, combination is the best. Missing data and artifacts, could be quite difficult for tools to overcome because it's identify things, right? So it doesn't have input on that. So it doesn't have data on that. So it could be some challenge. Limited biological understanding. Because this machine has a limited. If you don't have sufficient biological understanding, you don't know how to use the machine, how to correlate all of these things together. Technical. I think these fall under technical and statistical limitation. There's still not complete statistical models to tell the correlation between all of these. Some people might agree with that, some people might disagree with the statistical method they use in each of the experiment. And ethical and legal consideration, this one is mostly dealing about with the data protection. So let's say all of the private hospitals use this to predict your biomarkers and also a prognosis for you. You think it's dangerous? Yeah, because they can sell this data, then insurance don't want to assign your higher insurance rate for you, then usually I won't like that. So I think that's all from my presentation today. I hope you can understand what I'm trying to present. So that's all from me. Thank you.
We run out of time, so we'll move on to the next one. Well, we'll go back later today.
So no questions?
No.
Yeah.
Bring my laptop.
Oh yeah.
So excited. He doesn't have any questions.
Honestly, if you ask me any question, so I also should have have time to understand answer also. Because a lot of this related to statistics. And creating these algorithms.
Yeah.
那个大的
Very complex.
那个大的那个。
When I try to read the paper also, apa ini semua?
Yes.
But have you what's the size now?
Yeah, I think there are some models that they try to predict data. Because nowadays there are a lot of data, but normally they combine, yeah, combine. Also, there's a lot of noise, you don't know what is the real data and not the real.
My topic is application of bioinformatics in cancer diagnostics.
没有差几笔。
Use much earlier sequencing technology and response treatment methods. The five-year survival rate of breast cancer patients can reach 90%. However, there's still 30% of patients with recurrence and metastasis after initial radical mastectomy. While the survival rate of patients with advanced breast cancer is only about 30%. Therefore, the study of diagnostic markers is very important for the treatment of breast cancer. Tumor markers, specific molecules or proteins, are present in cancer patients. The clinical application involves in earlier cancer screening, diagnosis, and typing, treatment monitoring, diagnostic evaluation. The focus on reliable diagnostic methods is paramount for improving the breast cancers. The focus on identifying reliable prognostic markers is paramount for improving the survival rate. NGS is a high-throughput genome detection technology, which has retained between millions or billions of DNA fragments. And it also allows detection genomic or transcriptomic and epigenetic changes in tumor cells. Thereby identify potential tumor markers. And the NGS process involves sample collection, DNA extraction, library construction, and sequence and data analysis. Other advantages is high throughput sequencing, multi-marker analysis, and high sensitivity. And those have their disadvantages such as high cost and sample quality missing. And the challenges in bioinformatics first one is data complexity. The genome transcriptomic and the other data for tumor tissue, and complexity and the complex and diverse. Standardization, lack of on-field bioinformatics analyze standards. And the last one, sample heterogeneity. There's huge heterogeneity among tumor samples, including differences in cell types, mutation rates. And that's all.
Yes.
Thank you.
That's so there's no example.
This one is number.
I think the example is the first slide, right? But he didn't present the
Maybe
finding.
she's nervous. Are you nervous? Yeah, little bit, yeah. Okay. So don't worry, because I think you know everybody already. Yeah, you don't have to be nervous.
Represent investor.
Is that?
Maybe if you took too long, I didn't share. Okay. Yeah.
I'm Sultana and I'm going to present a case study on primary immunodeficiency. So primary immunodeficiency is a rare heterogeneous autosomal recessive disorder, and the authors here they used next-gen sequencing, single sequencing, and other bioinformatic tools to confirm the condition, genetic condition. So primary introduction, primary immunodeficiencies are rare heterogeneous group of autosomal recessive disorders that are characterized by an absent or impaired immune system. Sorry. I'm just nervous, sorry. Yeah, if there is a delayed diagnosis of PID, it increases the risk of morbidity and mortality of the patient. So the affected infants develop these infections during their first few months of age and generally die before 1 year of the age. So this condition is very rare. It's found in 1 in 100,000 newborns in the world. The patient history. Patient was a male infant born from non-consanguineous parents. Patient was normal at birth, but at 6 months of age presented with eczematous rash and a history of repeated infections. Initially, he was treated for seborrheic dermatitis. However, the rash was later determined to be due to low lymphocytic counts. He was readmitted at the age of 11 months with convulsions and altered sensorium. Patient had respiratory failure. So the blood profile showed low hemoglobin, low mean corpuscular hemoglobin, platelet, neutrophils, and monocytes. Flow cytometry tests reveal normal B cell counts, but less T and neutrophil cells and low levels of serum Igs. Next, this is a timeline of the patient. He initially he was having eczematous rash, which was detected as seborrheic dermatitis. He was treated for seborrheic dermatitis, but was not confirmed for PID. Then at 11 months, he had convulsions and altered sensorium, for which DNA sequencing was done, and he was waiting for the HLA match donor for bone marrow. And later he developed pneumonia and sepsis, and the patient died. Next. So the author here did exome sequencing and bioinformatic analysis. So the blood samples were collected from the patient and his parents. Genomic DNA was extracted from whole blood using KIAMP DNA blood mini kit from QIAGEN, and 3 micrograms of genomic DNA was used for sequencing, and the DNA library for exome sequencing was prepared using the Agilent Technologies Sure Select XT Human All Exon V5 kit. Next. Pay-dense sequencing was performed on the Illumina high sequencing 2500 Illumina with a read length of 100 base pairs for the whole exome using the True Sequence Cluster Kit V3. The analysis of the sequencing data, variant calling, and variant filtering were carried out. Through Sanger sequencing was performed to validate the IL2RG variant in the patient, because PID is most commonly caused because of IL2RG variant. Next. Results. So the whole exome sequencing of the patient with both of his parents resulted in an average of 96 million paired end reads. Approximately 96% reads were high quality and aligned to the reference genome. The genotype variants of the patient mother and father were subjected to joint variant calling using genome analysis toolkit. Okay. They did the sequencing and they analyzed the genome analysis toolkit and found that like 100,000 variants, out of which 17,000 variants were in the X chromosome. And next, they further analyzed through ANOVA and annotated the variants and found 60,000 exonic and 272 splice type and 500,000 intronic variants across the samples. Furthermore, common variants with minor allele frequency greater than 0.01 were removed after comparing the variants with common variants database such as 1000 Genome Project and PBS exome sequencing project and ESAC database. Filtering of all synonymous variants resulted in 1341 variants, out of which 1302 were exonic and 39 were splice-site variants. Next. So this is the workflow that they use genome analysis toolkit, and next they use ANOVA, and they removed minor allele frequencies, and after filtering everything, they got 1300 exonic and 39 splice site variants. Next. Discussion. So the authors noted that there is a potential causal variant, this variant, in exon 2 of the IL2RG gene located at X chromosome and location of the chromosome, the variant. This mutation replaces cytosine with adenine at position 252, resulting in an amino acid substitution of asparagine to lysine at position 84. Yeah, here is a mutation. Wait, this is the position of the mutation in the chromosome X, and this is a mutation where instead of G, there is a T. Yeah. Yeah, amino acid substitution of asparagine to lysine. The variant was hemizygous, as it was only found in the X chromosome and not in the Y chromosome. So it is heterozygous in the mother, obviously double X, and was not found in the father, confirming X-linked recessive mode of inheritance. Cytosequencing confirmed the nucleotide substitution. It's in figure this. So here it has to be C, but due to adenine substitution, it is T. Here we can see. The missense variant. Missense variant is found at the predicted glycosylation site located in the conserved extracellular domain. A multiple sequence alignment of the IL2RG protein with other species showed conservation of the asparagine residue, which was mutated in the patient. Figure 2b. Yeah. So this is the asparagine conserved region across the species. It is a conserved region, but it was mutated in the patient. Yeah. So authors also wanted to validate their findings, because the variant that they found, it is not found in the databases. It is not found in the human genome database. The variant that they found is a novel variant. So to validate that, they used sorts intolerant from tolerant. Now this predicts this predicts the function of the protein. If there is a substitution of an amino acid in place of other amino acid, it detects if the protein will function correctly or not. Next, they also use polyphen-2, which detects the substitution of the protein, resulting in difference in structure and function of the protein. Next, they also predicted disease-causing potential of the variant through mutation test. Next, they concurred in the prediction the effect of the mutation as potentially pathogenic, as this variant was not found in the common variant databases, including database SNP, 1000 Genomes Project, ESAC, and ABS. So here we can see through SIFT, they concluded that it is a deleterious effect. The variant had a deleterious effect and damaging and disease-causing potential. So yeah, conclusion, PID diseases are clinically and genetically heterogeneous, for which accurate diagnosis is essential for improving therapeutic outcomes. It is potentially fatal, with mutations in IL2RG gene being the most common cause. The next generation sequencing based clinical diagnosis can greatly increase the diagnostic yield because multiple genes are simultaneously tested. In this study, author identified a novel nucleotide change that resulted in an amino acid change of asparagine to lysine at position 84. But this amino acid substitution in the extracellular domain likely leads to an abnormal or unstable common gamma gene, which is heavily repeated in the protein, so thereby affecting signaling downstreams of several interleukins.
So, in such a case, maybe better to do prenatal screening for?
Yeah, because various tests that takes place prenatally, so it will be better if the next generation sequencing is also done prenatally.
But also the outcome is always
Unique.
almost straightforward.
So patient, we can either do it right or they and
It can be treated.
the symptoms can be managed. They can be heavily on antibiotics.
Oh,
They can undergo gene therapy.
okay. That is from a very after immediately after they are born, then they need to take some action. But also, because of course when the baby is born, they are very fragile and they can't undergo like major operations.
Yeah, but it's
At least something can be done.
yeah, it is this like if you do the NGS, it will just give us the time to plan.
Thank you. Don't have to be so normal. Yeah.
That's evening everyone. Evening. So
Good evening.
good evening.
I think that's the subtitle. I think the presentation you need to switch it up.
Good evening, everyone. I am Puspa, and I'm here to talk about how bioinformatics can be used for the diagnosis of autosomal recessive neurologic syndrome. Neurodevelopmental diseases, we all know it's a disorder which affects our central nervous system and brain. So these diseases are like clinically and genetically heterogeneous. So like it depends depending on the persons. So one of such disorder is autosomal recessive neurologic syndrome. It occurs when two defective genes from both of your parents inherited to you. Its characters are hypertonia, developmental delay, facial dysmorphism, and brain abnormalities. Case study. In this study, they took a boy, a 13-year-old boy, with a neurological disorder from an Iranian consanguineous family. Consanguineous meaning like the parents are related, cousin, they are from the same ancestors. So at birth, his growth parameters, it was normal. But then in his early infancy, like certain symptoms such as hypotonia, poor feeding, respiratory distress episodes, seizures, etc. was observed. And then after some time, signs of developmental delay and mental retardation was also observed. So, like, he was eventually admitted to the hospital. So on the side, his parents, they were like perfectly normal. They had no neurodevelopmental phenotypes, and in his family, like, there were no history of such symptoms. but his older sister had the same phenotype as him, and she had passed away after like few months of being born. So clinical diagnosis, after physical examination, it revealed that he had a developmental delay, epilepsy, tonic seizures, chest infections, and mental anomalies or brain abnormalities. And in his phenotypic appearance was like thick eyebrows, hypertelorism, sharp nose, thick lips, and all those mentioned here. And MRI of his brain revealed that like white matter atrophy in his parietal occipital lobes and short and thin corpus callosum. Then like he was like referred to for genetic evaluations. So this is the workflow of how they did this test, how they did his genetic evaluation. So once they generated the physical features and then samples, so obviously they extracted the DNA and then they like created the library and then aligned with the human genome GRCH38, and then they like did variant calling. It is the like identification of variants, and then did annotation, and then did the filtering of this variation based on parameters such as exonic regions, homozygous phenotype related genes, and they considered only those multiple allele frequency which were less than 1%, because like this condition is more like as I mentioned, it's an autonomous like recessive inheritance thing. And then after filtering the variants, they evaluated those variants based on like clinical significance, just to make sure that like what this what is the connection of these variants with the various diseases. So genetic findings. So like they evaluated or sequenced their exome, whole exome. So after sequencing the exome and then like aligning with the human genome and doing the variant calling and then annotations, they like, so these were the different types of variants that they obtained at different stages. So here, after annotation, they chose homozygote variants as I mentioned, because as I mentioned earlier, it's a recessive inheritance thing. So after this, they like filtered the variants using different parameters. These are the parameters, and then at last there were like 19 variants, and these 19 variants they evaluated based on the like clinical significance and in silico pathogenicity. So finally, at last, they just found one variant, which is the possible cause for this disorder. So what they found was like they found that there was a mutation in protein phosphatase 1 regulatory subunit 21. So this is a diagram. So this is the chromosome, and here you can see that this is exon 12, 13, 14. So what they found was there was a mutation in the exon 13. So there was this PPP1R, actually it's 21. Okay, it's located in exon 13, and there's a mutation there. So what they found out was there was a deletion of 2 nucleotides, AG, in this gene. Here, these are the genes, conserved genes found in different species, and here's the human, and this is for the patient. So as you can see here, like this white thing, it's all similar in all the species, including human, but in the patient, this is like mutated into a stop codon. So also they verified this with Sanger sequencing. This is the sequencing of father, mother, and the son. So father and mother, they just have one, like they're only one AG is deleted, like here. So like, but in the son, like you can see, like both of these are deleted. It's just there is no T. It's just C. So like Sanger sequencing confirmed that it's a homozygous mutation. This is the pedigree of a family. So as I said earlier, this is caused when two mutated genes or deleted genes are inherited from the parents. This is the son. So this is parents. So they have one mutated gene AG, but when it comes to the son, like both of them are gone missing. So because of this, the son has this disorder. And another thing about this protein is that like this PP protein phosphate 1 regulatory subunit 21, it interacts with protein phosphate 1 protein, which is like responsible for carrying out different cellular functions. So what happened is that after due to this mutation here, so like the premature stop codon is introduced. Because of these non-functional proteins produced, as a result, this final product of this gene becomes non-functional, and then the protein phosphate 1 regulatory protein, it cannot interact with the protein phosphate 1. So as well as it's all stopped there. So also what they did was they performed this string test, or let's say this database. They looked into this, and what they found is that this PPP1 to R21 protein, it had a strong association, or let's say networks, with other genes. These are the different other genes that are responsible for like this disorder, neural disorder. So it's just which further gives us more information that okay, it's true, what they were doing is true, and the result is true. So conclusion. So despite technologies being developed and developing and improving, there was still some gap in the detection of such kind of mutations. However, with NGS, it can be NGS being able to sequence the proteins or the varieties of the proteins in these neurodevelopmental disorders, can improve the sensitivity of mutation detection. It can help in invasive or prenatal screening, and then also identify rare genetic variations. And using these results, we can these results can be also used to genetic counseling to families with high risk of intellectual disabilities. Yeah, thank you.
I think the presentation is very clear, so at least for me, I can understand most of it. So she will explain about certain terms that were used, like sanguinal family. Yeah, that's very important to understand the whole story. So if no questions, then we move to the last presentation. We will record and send. Yeah.
他没有，
到你了。
他没有。
Mr.
Yeah.
Yeah.
差不了一点，只漏完话
照片就好。那你的那个有时候
插进去吗？ 这个念就好，这个念去写。这日子是真难呐、照着念就好了，就念，念就有分了。我跟你说，你看到一个字儿一个字儿蹦的时候，你就这个念不出来了。OK，好，这什么玩意儿？哎，是这个样子吗
Symptomatic pig nut allergy. 哇，我都 stat 里去了。
Mental fatigue is bioinformatic about peanut allergy. The peanut allergy is type 1 hypersensitivity. B cells are stimulated by CD4 cells to produce IgE antibodies specific to an antigen. During sensitization, the IgE antibodies bind to FC1 receptors on the surface of tissue, mast cells, and blood basophil. Mast cell and basophils coated by IgE antibodies are sensitive cytokines were released. We chose 32 children and adolescents. All study subjects had an evaluation that consisted of an extensive history, physical examination, SPT, and measurement of serum peanut SIG levels. We use the UNICAP system to determine for each sample the SIG level, and use the ISS to determine the peanut component allergens, ARH1-2-3, and then scanned by using the scan array, to make a picture. the microarray was blocked with the BSA and followed by the addition of OGT. And FICT conjugated antibody. The enzymatic reaction was run by pumping the mixture up and down through the porous A1203 triple material. Image of the fluorescent signals were generated every 10 minutes during 4 hours. Images were quantified using the BioNavigator 6 software. Uh, 31 patients reacted to peanut. Defined as a peanut allergic group, and 31 did not react, defined as peanut tolerant group. 13 of the 31 peanut tolerant patients had a convincing history of previous acute reaction following peanut consumption, but they apparently outgrew their allergy. The remaining 18 patients had positive peanut SLGE levels. The 16 is positive, but never had clinical reaction following peanut consumption. Peanut allergic individuals had significantly larger wheel diameters than did peanut intolerant subjects, but their peanut IgE levels were not significantly different. Peanut allergic individuals showed a significantly more intense IgE binding to peanut epitopes than did peanut tolerant ones. 31 IgE binding regions containing multiple epitopes were identified by using tile maps. the developed decision trees are one of the end products of machine learning analysis is shown in picture 3. 62 individuals were sorted on the basis of their Ig reactions to the selected peptide biomarkers. 30 of 31 peanut allergic and 29 of 31 peanut tolerant individuals could be correctly identified by using this decision tree. The diagnostic performance of this allergy test method was compared with that of other methods, SPT and UniCap, by using ROC cables in picture 4, which displays the relationship between sensitivity and specificity at different thresholds. The area under the ROC cable indicates the diagnostic performance of the test methods. The greater the area under the curve, the more precise the method. Currently, the most definitive diagnostic test for peanut allergy is the DBP-CFC. However, the complexity and the potential of DBP-CFC to case threatening allergic reactions after affects its clinical application. We use TileMap, a bioinformatic software to identify informative epitopes by comparing the peanut allergic individuals with both peanut tolerant and negative individuals. TileMap was developed for DNA tiling microarray to identify binding sites of transcription factors across the genome by using overlapping probes. it is more advanced and sensitive than other commonly used studies. Statistical tests because it takes into account the overlapping feature of the spotted groups and significantly increased the sensitivity of the analysis. The machine learning method, another bioinformatic tool was applied to develop prediction models and identify the combination of peptide biomarkers that maximize the diagnostic power. Fivefold cause validation was used to avoid overfitting the prediction model. Thank you.
Any questions for Carl? No.
直接拔了。像你吃饭，你不能说哈。你放这就好了，放这就好了，放这就好了，放在那边就好了。他把你过。
So this is the last session for DFM 5 to 4. So you continue with the other classes.
Yeah, and type of reach.
Yeah, what to do, and also is there any more?
Oh. I mean, 501, that was already finished.
No.
Yeah.
I don't think so. It's one bit like so that is obsession.
Oh, okay. Alright. So the quiz will be on the 29th, as we discussed just now. We open the full day. And then for the presentations, I will set up a folder in the email so you can upload the presentation file. Yeah. Already there, right?
Yeah.
Okay. Did you upload the paper also that you use as a reference? Yeah. Go together with your file.
Only the mains.
Yeah. I mean the main. Yeah, so yeah, so that's it. So if you have any questions, you can email me or see me. But next month, yeah, I have another class for 5 to 2.
We'd love to see you.
Told me the last class already.
Yeah, it's for 5 to 4. The other topic is for 5 to 2.
I have like,
Yeah. What?
the protein talking that we didn't learn is actually quite important. That we don't have a health as protein talk is actually quite.
Oh, really? Which one? Which topic? The last one.
Chapter 5.
Chapter 5.
Because I thought in that module itself, although it has like the chunk.
Oh, okay. previous year's comments, then we kind of sometimes we say, okay, we need to do more on this area, then we will add more. When we shouldn't like
That's like so like food, right? And is there anything commonly used here?
yeah, the research projects, the research projects we do a lot of for my case, we do a lot of deep learning services, use the genetic information of genes, so you can CR and for protein expression. Along with like overall, you need a lot of these of course, structural modeling. So although it's only using computer, there's some part of whether so you need to check the structure of the computer and then try to modify the enzyme and that. Yeah. Another thing also we have to do antibody work. So look at combinations, right? We have antibody screening. So screening sometimes we need to so you need to know this. So you need to do some kind of that even though it's like majority when that you still need to do some kind of computer work to analyze your data or how to experiment.
Any X-ray study on Friday?
Yes.
X-ray study on Friday.
Yeah, I think Dr. Tom will be the closer. Yeah. But maybe 10% that I think. We drive that later on that. Yeah, already done the part. And do the message and get the yeah, so you have like some kind of partner on that, then you could really do that.
So the aspect of bioinformatics involved in molecular method is actually mostly doing sequencing.
Yeah, so it helps us in knowing the sequences. Some people are doing transcriptomics for cancer research like Rockbridge. So there are different aspects. Well, let's take a photo for the last class.
So yeah, project available.
Where shall we switch here?
Or is there any website
Maybe for the final year. Okay, it's gone down. 4, 3, 2, 1. So far, yeah. What more? 5, 4, 3, 2, yeah.
Thank you. I want to see ahead of me. Quite ahead of coming out.这不包你的包围，只可以包吃包住。对呀、你把我印象给我，你什么不会吗？啊，你去过深圳？知道。我想哦，才不会啊！啊，是现在是什么？时候就是到一晚了，这个可以看。我手机今天是有火车拉给我去来了，但是后这场是我自己去的，就是为什么要我给我的话，我就没有这个，但是每次要但是这个就有刷了是吧？就是杀还是个杀了？杀，这外国人是杀一不杀，杀以上了，是不是？多，这边的 Jack 都没有到100多的，那4杀了。是，这是为什么很贵啊？这个地方，除非你住那个好像什么一样的，住那种通风啊，住到住那种楼嘛什么这样子，那种就便宜，但是也是便宜，是200多吗？
哦，limit 跟 maximum 是一样的。limit 跟 maximum。图书馆。这是所有的对吗？哦，这个是啊。哦。然后这么多，哇，这个也写哦。今天。所以知道我们家了。哦，有这个哦。难怪这样好了，手臂手。谢谢你。等等 OK。
我跟你讲，你要不是我不好行动，
最好的，最好的人都是美的。我们这个是不是等到明天才会有？是那个考试的。那个考试。哦。我记得今天有课，可以要去看一眼嘛，看实体，可以多抓点。办公室。对呀，他是个程序员，还好吧？我觉得。
我就喜欢那样的。
一周看。周还是别人你没有在看节目的。我很好看。是吧？之前不是跑了没抓到那个补铁团追过他车，追尾是什么？我说之前好像不是有过他车追尾。那你刚才为什么问深圳比香港便宜还是贵？想说，他说深圳比香港便宜，那说深圳比香港便宜，便宜很多。那我觉得深圳跟香港差不多。是不是一样吗？我跟你说，我在深圳的那个市中心的中心，我可以住到200块钱。啊！你去九龙，你去住200块钱看看。
不是，就除去酒店，我知道酒店，除去酒店呢？
吃饭也贵。吃饭我觉得香港还可以啊。他是那个，我记得云吞面差不多但是我记得还要30多、40多块。没有看到。
全部限制了我们的想象。如果我去香港是，我会选择，然后随便找到就巴黎。那我去做深圳，我可以做经济家，就是有点麻烦，因为我只要是回深圳，然后再去香港的话，要浪费一次考证。那你是去做完也是有路。不但因为我们中转的话是可以拿护照直接进香港，但是如果你从大陆再回香港的话，你就要加2年。
那个4度多少钱？每年？
你自己签吗？不是，其实可以无限签，就是你每回去至少可以再签一次啊。现在是可以无限签。这就是我们讲的，比如说他也是从深圳来的，可是应该我也很久。那时候我亲戚家在南城那里，算是比较远一点吧，比市中心远一点。你老家能30去。他这样多少？他是建农的。啊？
哦，房地产的。
房地产。我亲戚是建农。对啊，所以我说可以去帮你测好，你住1个星期都没有问题。我要刚说一声而已。然后每一次都吃的，每一次都吃的很好。你们内部价买房子？啊？你们内部价买房子？这个好像可以吧？
内部价你买得起吗？
这个我不知道。给你10%，连我干嘛？
啊？你10%吗？这么夸张？
那印象不太懂。那个
印象不大呀。
You walk into the.
What's wrong with walking?
It's very far.
No.
Don't know this.
Most family.
他们要去哪？
啊，他们要去什么晚？
对。
现在是图书馆。
拜拜。
不知道啊，
不是图书馆啊，那是图书馆。
我不知道啊，因为我这边跑出去了。